Hello everyone, welcome to the new video tutorial of Bio and Tech. In the last session we have discussed about few types of microscope. In this session we will be discussing the third part of microscopy. If you haven't subscribed to our channel, subscribe to it and enable the bell icon to get the notification. So without wasting time, let's get on with the video. So in the past classes we have discussed about 6 types of microscope. In this session we are dealing with the 7th type of microscope, that is fluorescent microscope. The technique is known as fluorescent microscopy. And the principle of this microscopy is that certain substance, certain chemical substance or whatever it may be, maybe sometimes it can be cell organelles, so whatever it may be, it absorbs the UV light and re-emits a visible light. So that phenomenon is known as fluorescence. So the principle is that it absorbs the object we want to observe, or a certain chemical compounds absorb UV light and re-emit visible light which can be viewed through our eyes. That's the basic principle of fluorescent microscope. But what happens is that most of these biological specimens do not have the capacity to this property. So what happens is that we add certain chemical compounds which has the ability to absorb UV light and re-emit visible light. So that compounds or that substance that we add or tag with the biological specimen is known as fluorochrome, that is a dye. So but certain other cell organelles, like for example chlorophyll, have the ability to absorb UV light and re-emit the visible light. So that substance or that particle is known as autofluorescent. The basic application of fluorescent microscope is in the diagnosis field. It is also used in immunofluorescent techniques. So that is the basic concept of fluorescent microscopy. The 8th type of microscope is electron microscope, which is a very familiar one. It is an important discovery in the field of science. The two scientists who developed electron microscope is Noll and Resker. This name shouldn't be forgotten. It might be asked in the entrance exam. So Noll and Resker shouldn't be forgotten. You should note it down. So in the electron microscope, what difference is that in the ordinary microscope we use light as a source, but in the electron microscope electron beams are used. And instead of lenses in the ordinary microscope, we use here electromagnetic coils. So that is the basic difference between electron microscope and the ordinary light microscope. So that's the two difference. And the wavelength of light is much higher, but compared to the electron beam, the wavelength is very much lower. So you can note down the equation, that is that is a limit of resolution is equal to 0.61 lambda by Na. And as I have mentioned before, numerical aperture is almost constant for most of the microscope. The only variable that we can change is lambda, that is the wavelength of light. So if you use a wavelength which is very small, the limit of resolution will be low. That means resolving power is very high. So that is the basic concept or principle of electron microscope. So two types of electron microscope are there. One is transmission electron microscope and another one is scanning electron microscope. Now let's see the two types of electron microscopes. Okay, now the first type of electron microscope, that is a transmission electron microscope, or otherwise TEM. It is actually used for understanding the ultrastructure of a cell or biological material. For example, taking the case of bacteria, to understand the ultrastructure of bacteria, transmission electron microscope, otherwise TEM, is used. Now first let's understand the basic structure or components of a electron microscope in a simple way. The first one is an electron gun, which is a V-shaped coil and it shoots the electron when it is heated up to a high temperature. Then there is a cathode ray tube which focuses the electron, and a condenser coil which focuses the electrons to the object, and from the object the image is collected by objective coil and passes to the projector coil which projects the image in a view plate. So that is the basic component or structure of electron microscope, both in the case of TEM and SEM. But this the positioning and different slight modifications are there, but the basic components of electron microscope in TEM and SEM are same. So that's the basic application. As I mentioned before, the viewing the ultrastructure of a cell or biological material, TEM is used. And apart from that, there's an important point that the living objects cannot be seen with the TEM. That is the basic point you should understand. The living objects cannot be seen with transmission electron microscope. So that you note it down, because living objects, the live objects cannot be seen. Only dead objects can be, or dead biological material can be seen with the help of transmission electron microscope. Now let's see scanning electron microscope. So the next type of electron microscope is scanning electron microscope, otherwise SEM. So it is actually used for the surface view, to understand the surface structure of a biological specimen or any other material. This scanning electron microscope is used. The basic principle is that once electrons are shooted, secondary electrons reflected back is used to analyze surface structure of of specimen in scanning electron microscope, whereas in the case of transmission electron microscope, the electrons pass through the object, living object, and that strikes the viewing plate, and that forms a characteristic image of the inner ultrastructure of a biological material. But whereas in the case of scanning electron microscope, the electrons are struck, and when a secondary electron is reflected back, that is used for building a three-dimensional image. That's a major advantage, a three-dimensional image of biological specimen or any other compound that is used to view in an electron microscope. And another advantage of scanning electron microscope is that a whole specimen can be used to understand the structure. A whole specimen can be used. So that's a major advantage of scanning electron microscope. So that's all about electron microscope. Okay, and the final one is confocal microscope. The scientist who developed the confocal microscope is Marvin Minsky. So the name should be forgotten, it should be noted down. Marvin Minsky is the person who developed confocal microscope. Now the advantage of confocal microscope when compared to normal microscope is that it has shallow depth field, and the elimination of outer focus glare. That means the image that we get with the help of confocal microscope will be much higher in contrast and much highly visible than compared to the ordinary microscope. And moreover, a three-dimensional image, almost a three-dimensional image, can be obtained through confocal microscope. And another advantage is it has the ability to collect serial optical section from thick specimen too. So from thick specimen also we can get a highly efficient image when compared to a normal ordinary compound microscope. But the major application is that we can get a three-dimensional image, and another application is that live specimen can be viewed through the confocal microscope. So that's the brief outline of confocal microscope. So that's all about what we have discussed about microscope. If you have any doubt or confusion, please mail it to bioantag@gmail.com. So wait till next video. Bye.